Hello everyone, in this video we will be discussing the principles of loose connective tissue structure in less than 10 minutes. Connective tissue comprises a diverse group of cells within a tissue-specific extracellular matrix. In general, connective tissue consists of cells and extracellular matrix. Extracellular matrix includes structural components like fibers and specialized proteins that constitute the ground substance. So we have cells, extracellular matrix. In extracellular matrix, we have fibers and ground substance. And different types of connective tissues are responsible for a variety of functions, including homeostasis, mechanical support, and maintaining internal environment for many organs. There is a big classification of connective tissues in human body, but today we are focusing on connective tissue proper. And this family of connective tissue proper has two representatives: loose connective tissue and dense connective tissue. Both of this type have the same structural components like cells, fibers, and ground substance. But if we talk about loose connective tissue, cells predominate. If we talk about dense connective tissue, fibers predominate over other compartments. Extracellular matrix is a complex and intricate structural network that surrounds and supports cells within the connective tissue and also creates an environment for their communication and movement if the cells are mobile. Besides fibers, extracellular matrix contains a variety of proteoglycans, multi-adhesive glycoproteins such as fibronectin and lamellin, and glycosaminoglycans. Three types of glycosaminoglycans, as dermatan sulfate, keratan sulfate, and hyaluronan, constitute the ground substance that is the part of extracellular matrix in loose connective tissue. There are three types of fibers that are commonly found in connective tissue. The first and the most commonly found is collagen fibers. This fiber is responsible for the strength of the tissue. There are over 20 types of collagen fibers in our body, depending on their localization in specific tissue or organ. Then there goes elastic fibers. So by the name, we know that this can change their shape and present in elastic cartilages and in structures that are flexible. And the last type is the reticular fiber. This fiber loves to be stained with silver. That's why it is called argyrophilic, and it is found in lymphatic organs responsible for the production of blood cells. The synthesis of collagen fibers is done by fibroblast, and there are two stages: intracellular and extracellular. In the intracellular stage, we have the production of alpha 1 and alpha 2 chains. Then we have a specific sequence for them. We create triple helix, and this triple helix is exocytosed in extracellular matrix, and all of the further three steps are happening outside of fibroblasts. And in the extracellular matrix, what happens next? Our collagen molecules assembly, we create collagen microfibril, and from microfibril, we create collagen fibers that are ready to go into specific localizations depending on their type. Now let's move on to cells. When talking about connective tissue, it is important to understand that there are two populations of cells: resident cell population and wandering cell population. So as for the residents, this includes fibroblasts and fibrocytes, myofibroblasts, macrophages, adipocytes, mast cells, and adult stem cells. As for wandering cell populations, these are those which may be present in the tissue itself or into the circulation. So this includes lymphocytes, plasma cells, neutrophils, eosinophils, basophils, and monocytes. So the most important here is fibroblast because it is super active synthetically, and fibroblast on his own can create all components of extracellular matrix, including ground substance and fibers. Fibroblast is spindle-shaped, elongated, and if it lacks its synthetic activity, it becomes definite form, and such cell will be called fibrocytes. Also, fibroblasts can create myofibroblast. So this is a cell that is something in between smooth myocytes and fibroblasts, and often found in the regions where our, for example, skin or loose connective tissue is reconstructed and creates a scar. So the cell line of fibroblast, which includes fibroblast, the actively synthesizing cell, fibrocytes, its definitive form, which is low in synthetic activity, fibroclast, the cell that lyses extracellular matrix, so cleans up, and myofibroblast, the intermediate cell between smooth myocytes and fibroblast itself. So all these four cells create the fibroblastic differon, or the fibroblastic cell line that is the main in the composition of connective tissue. Besides, fibroblasts are also called melanocytes because together with fibers, these cells provide mechanical strength to the tissue, and they produce a lot of growth factors that are important during differentiation of embryonic tissue. Myofibroblasts are isolated cells. They come from fibroblasts, but in contrary to fibroblasts, myofibroblasts possess actin filaments with associated actin motor proteins such as non-muscle myosin that enables them to contract, and they're rarely commonly found in the myometrium during pregnancy. Next, we will be talking about the cells that are derived from the blood cells, but they live in connective tissue. And first on the list is macrophage, or we call them histiocytes. These are the cells that are derived from blood cells called monocytes, and they possess a very high phagocytotic activity. That means they can eat up foreign particles, bacteria, etc. Therefore, in their cytoplasm, they will have a lot of lysosomes, Golgi apparatus, mitochondria, and secretory vesicles. What is important that there are type 1 macrophages, type 2 macrophages, depending on which function predominates in them. But when macrophages encounter large foreign bodies, they may fuse to form large cell with as many as 100 nuclei or more that engulfs foreign body. And these multinucleated cells are called foreign body giant cells or Langhans cells, and they're really important in pathological diagnostic. Next, we have mast cells. They develop in the bone marrow and differentiate in connective tissue. They are large ovoid connective tissue cells with spherical nucleus and huge granules that possess heparin. They arise from pluripotential hematopoietic stem cells, circulate in the blood, and then move to the connective tissue to become mature and stay here in need of allergic or immune reactions. And at this point, you may say, okay, we have really similar cell, which is basophil and also has histamine granules. And are they the same? The answer is not, they're not. Even though mast cells and basophil come from the bone marrow, they have different stem cells precursors. And mast cell circulates in the blood for a very short time and stays in the loose connective tissue and degranulates here, whatever basophil works and spends all of his lifespan mostly in the bloodstream. And as you see from the table, the priming factors and mediators are actually different for both of the cells. Then we have plasma cell. This cell is derived from B lymphocytes. It is specialized in production of antibodies that therefore it is working for humoral immunity, and it has very specific morphology. So as you see from the scanning electronic microphotography, the nucleus of plasma cell is always shifted aside. So it is acentrally located. And there is a very light space straight in front of it. And basically this is where complex of Golgi is located. And we have abundance of rough ER. And the last two cells that we will discuss will be adipocyte and pigment cells. So adipocytes, in case this is a white adipose tissue, each cell contains one droplet of fat, and this droplet pushes on the nucleus. That's why it is really flattened also on the side of the cell, and it creates an impression of a signet ring morphology, and it is very commonly described for our white adipocytes, whereas in brown adipocytes that is found for newborns, for example, we have many droplets of fat and they're not pushing on the nucleus and it will be of typical morphology. And pigment cells or melanocytes that derive from neural crest, it is important because the tumors that will be developing from melanocytes are really aggressive because of their embryological origin, and they produce melanin in the form of melanosomes that is stored in their cytoplasm. So that was it about how loose connective tissue is organized, and about tumors and pathology and more detailed physiology of connective tissue we will be discussing in next videos. Thank you for watching and see you soon.